Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Nanopore Sequencing for RNA Vaccines and Therapeutics, Advancing Drug Development and Quality Control. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions or comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, which is found on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the comments tab. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming to those who register on xtalks.com. At this point, I'd like to thank Exotera, who developed the content for this presentation. Exotera is a specialized CDMO partner delivering preclinical and clinical process development, manufacturing optimization, and CMC support for virus and RNA-based therapeutics and vaccines. They assist clients from start to scale up to the commercial manufacturing, including analytical testing and regulatory consultation. Thanks to their optimal technologies and experienced team, they help bring innovative therapies from bench to patient with quality at speed and cost effectiveness. Now it's my pleasure to introduce our speakers for today's webinar. And first, I'd like to introduce you to Mikael Lambin. He's a development tech lead manager at Exotera. There he is. Hi, Mikael. He is an accomplished leader in biotechnology and pharmaceutical quality management with over 15 years of expertise in analytical method validation, process improvement, and team leadership. With a PhD in molecular biology, Mikael has contributed significantly to scientific research and global QC advancement at GSK before joining Exotera. So there he is. Welcome. Now I'll welcome our second speaker, and he is Dr. Olivier Lucas. There he is. Hi, Olivier. He is the Director of Biopharma, Europe, Middle East, Africa, and India at Oxford Nanopore Technologies. He previously held different clinical and oncology leadership roles at Oxford Nanopore. He has a background in molecular biology and has been in the genomics industry for over 12 years. And next is Dr. Wei Wang. She is a senior analytical scientist with a PhD in molecular biology and 13 years of experience in sequencing technologies, including Nanopore, NGS, and Sanger. So there she is, Hi Wei. Leading the sequencing platform in both R&D and QC, Wei is specializing in long short reads, sequencing, assay development, and qualification, with expertise in Oxford Nanopore Technologies for analytical assay development for both nucleic acid and viral vector projects, NGS-based DNA, RNA virus sequencing, and bioinformatic pipeline development. Wei brings a deep understanding of sequencing methodologies in the biopharmaceutical industry. So those are our speakers. Now, before I pass the mic over to our first speaker, we do have a poll question for our audience members to engage in. And this is done in real time, and I'm going to launch it now. And you should see it appear on your screen. And the question we have for you is, how do you currently handle RNA analytics in your organization? Is it we outsource to a CDMO as part of a broader manufacturing project, or we outsource RNA analytics as a standalone service, or is it we handle analytics in-house and use Oxford Nanopore Technologies, or we handle analytics in-house using other sequencing analytical platforms, and finally, we are still exploring our options. So please go ahead, everyone, and cast your vote. I'll give you about 30 seconds to complete this poll question. Then I will close the polls, share the results, and then we'll get right into this great discussion with Mikael. Okay, and that looks like it is the majority of our audience. And okay, now I'm going to close the poll. So let's take a look at the results. Okay, here are the results. We have 0% say we outsource to a CDMO. Then we have 0% again for we outsource RNA analytics as a standalone service. And then we have 26% who said we handle analytics in-house and use Oxford Nanopore Technologies. And then finally, we have 16% who say we handle analytics in-house using other sequencing analytics platforms, and that's analytical platforms. And then finally, wow, 58% said we are still exploring our options. So thank you very much, everyone, for participating in that poll question. Now I'm just going to pass the mic over to Mikel. So Mikel, when you're ready, you may begin.
Thank you for the nice introduction, Sonia, and very interesting results from the poll. Hello, everyone. My name is Michael Landin. I'm the technical lead manager here at Exothera, located in the south part of Belgium. Before I hand over to Dr. Olivier Lucas and Dr. Wei Rong, I'd like to briefly introduce Exothera and the services we offer. So Exothera is an end-to-end CDMO partner driven by the purpose of manufacturing biotech drugs faster and more efficiently so that everyone, everywhere can get them. We specialize in the development and production of medicines based on viral vectors and RNA, both messenger RNA and self-amplifying RNA too, covering projects from prophylactic, personalized cancer, and even animal health vaccines. We also have a large portfolio of in-house analytical methods for characterizing our customers' product, as well as a deep expertise in analytical development. These analytics can be offered as a standalone service if requested. I encourage you to check out our website for more information on our services and technology. For today, let's focus on the RNA expertise and services we offer at Exothera. So, as mentioned, Exothera is an end-to-end partner offering a comprehensive list of services so that our customers can find everything they need under one roof for their RNA projects. We can support our customers at every step of their product's life cycle, from early R&D to clinical GMP, sorry, and full-scale manufacturing to commercial manufacturing too. On the next slide, please. So what really sets Exothera apart is our standardized RNA production platform called Intensify, optimized by our sister company Quantum. This platform includes a unique and optimized RNA production process, along with a range of equipment to cover all quality ranges of purified RNA from R&D to GMP grade. Some of these equipments are fully automated, as the one you can see in the lower part of this slide on the right. It's as simple as feeding the equipment with your specific linear DNA template, pressing a button, and the equipment will autonomously perform the IVT and purification of the RNA. The key to this optimized process is that the RNA is purified in one single step using magnetic silica beads, which is quite unique in the industry. Next slide, please. So the same process is used regardless of the sequence and quantity of RNA to be produced. A positive outcome of this approach is that it allows us to use pre-made reagents premixes. Since we always use the same process with the same reagents and materials, batches of reagents premixes, both R&D and GMP grade, can be produced in advance and be readily available as soon as a new project starts or when a production run must be performed. We produce, by the way, and release these premixes ourselves at Exothera. Next slide, please. Depending on the grade and the quantity of RNA, needed, required, or standardized process can be performed at different scales with equivalent final RNA quality. For R&D work, we run IVT, so in vitro transcriptions, from less than a milliliter, around 100 microliter, up to 200 milliliters, producing from approximately half a milligram up to 1 gram of purified mRNA. For GMP batches, we can produce from 100 milligram up to 3 to 4 gram of purified RNA, and we'll soon have the capability to perform 3.2 liters of IVT in one single production run on our automated GMP equipment to produce more than 12 grams or more of purified RNA in one batch. So let's talk about the scalability of our process. In fact, we do not scale up, we scale out. So from the IVT volume of 20 milliliter, we multiply this volume to reach the IVT volume we need. For example, to produce a batch of around 1 gram of RNA, we would engage 10 times 20 milliliter of IVT in one run. This allows us to produce RNA of identical quality from microliter, milliliter scale up to liter scale. Together, the standardized platform and scale-out approach we use save considerable amount of time for our customers' projects, as no process development is required, in addition to considerably de-risking their project, as they know the quality of the product very soon after the start of the project. So we produce small batch, small scale, you would have the final, the same quality at larger scale for your GMP production, for example. Next slide, please. So to summarize, Exotera is a CDMO partner offering all mRNA manufacturing steps under one single roof, including production of mRNAs from linearized plasmid DNA, their encapsulation and formulation, as well as fill and finish of the final product. We use a one-fit-for-all process and automated equipment for mRNA production that we scale out to produce the quantity needed from R&D to GMP production. Before passing the talk to my colleagues, and you probably already very well know all of this, but I just mentioned that mRNA vaccines produced in vitro mimic natural mRNA. They have a 5' cap, 5' and 3' UTRs that stabilize the transcripts and enable translation. A coding region that can include anything, antigen, antibodies, protein replacement therapies, etc. And also a poly-A tail. On the next slide, please. And so to ensure the safety and efficacy of mRNAs produced in vitro from linearized plasmid DNA, understanding whether everything went well during the various process steps is extremely important. This has led the USP to come up with a set of guidelines for characterizing mRNA vaccines. The testing panel you see here is only a subset of what's required, but you can see it's fairly intensive and very time consuming to do. It's very time consuming to do all this testing. Today, my colleague will present how Exotera uses Oxford Nanopore sequencing to try and streamline this testing that requires a large number of devices, in addition to be time consuming. But without further ado, it's my pleasure to pass this talk on to Dr. Olivier Lucas from Oxford Nanopore Technologies. Thank you for joining me today.
Okay, well, thank you, Miguel. I'm going to launch our second poll question before I pass it over to Olivier. So here is our poll question. And there you go. In your opinion, what is the most valuable aspect of real-time sequencing technology? So we have four options below. Is it speed and real-time data generation, or long read capabilities, or is it flexibility in sample types, or simplified library prep and workflows? So please go ahead, everyone, and cast your vote. Just like before, it's done in real time, so your participation is strongly encouraged and very much appreciated. Okay, so it looks like we have not quite half of our audience participating. So if I can get everyone now just to go ahead and make your selection from one of the below. Okay, great. Now we got half of our audience already selected. If we can get the other half to participate now. Oh, great. Thank you so much, everyone. All right. Okay, that looks like it's the majority of everyone. Okay, I'm now going to close the polls. And let's take a look at the results. Okay, so here are the results. 15% of our audience said speed and real-time data generation, while 52% of our audience chose long read capabilities, 15% said flexibility in sample types, and then 19% said simplified library prep and workflows. So thank you very much, everyone, for participating in that poll. And now I'm going to pass the mic and the controls over to Olivier. So Olivier, when you're ready, you may begin.
All right. Thank you very much, Nadia. So I'm very pleased by the answer that you shared with us today in terms of the long read, but also a lot of you do value the other capability that Nanopore brings. And today I will tell you more about the solution that we have at Oxford Nanopore specifically for the biopharma industry. So Oxford Nanopore Technologies is a global partner now present across the globe, originating in the UK, over 1,200 employees nowadays. We have a very strong customer support that is also available across the globe. The platform that we have are very modular, and as you can see on the top right, they go from fairly small, fitting in the palm of your hand, to more desktop-size devices. So we started to look into the biopharma market a few years ago. We really started to look at it from the angles of the R&D, which was some of our core market in terms of biomarker discovery, and started to peek into the development clinical trials, but also the manufacturing aspect of biopharma. And that was really highlighted by the mRNA vaccine explosion through COVID. And what we have realized at that time is Nanopore has a real true value proposition for quality control, specifically for mRNA, but overall in biomanufacturing. And that started the method development all the way to basically enable bulk release quality control. But that's require GMP grade solution. So we've been working at that very heavily with a very deep commitment within Oxford Nanopore over the last few years. So indeed, we did want to enable the market with validated method for the GMP environment. But for that, we felt like we needed to work with the analytical development team and maybe the method development team. And we started to do that over the last 2 years. And now we have a whole portfolio of solutions that is either available for evaluation for plasmid identity testing or mRNA identity with integrity and purity attributes coming up soon. But we've also developed other kind of assays and are in the process of validating them prior to commercialization in terms of integration site analysis, AVA and AAV testing also. So you may wonder, how does nanopore sequencing work? As shown on the video on the left, you see a double-stranded DNA, but it could be a single-stranded mRNA that is fed through a nanopore in blue. And the nanopore is a biological protein embedded into a membrane. And that membrane is really not permeable to ion, so to electrical current. Therefore, if we apply an electrical current, the DNA being negatively charged, it will flow through the nanopore, as shown on the right-hand side. And it flows at a very specific speed that is controlled by the motor enzyme in purple. As the DNA flows through the nanopore, you can see that there is an electrical signal change in blue, the moving wave on the right-hand side. And that signal, that electrical signal, is then base called, so transformed into a DNA sequence. So what does that mean for you for the application you may want to have within the biopharma world? First, we have access to direct sequencing, meaning that we do not need to do PCR. We do not need to amplify. And that means that for mRNA, we can sequence native mRNA without having to go to cDNA. And therefore, we have access to modified bases like the N1-MPU. Now, as some of you did answer in the poll, it is read length agnostic, so we can sequence really long molecules, kilobases, dozens, hundreds of kilobases long. As shown on the right-hand side, the data comes in real time. You launch a run, and within a matter of minutes, hours, the assay is completed. The chemistry is fairly simple, which enables quick workflows that are scalable, that can be basically highly decentralized into factories or near samples. So what we have done is we have packaged a solution into a new device, not one of the research devices that we have, but a new device that is 21 CFR Part 11 compliant. It's called Gridiron Q, with the intent to be used in GMP facilities. It is a lockdown version of the devices that we have, and there is a very clear commitment in terms of any kind of change that won't happen for at least 12 months. So meaning when you validate something, it's going to be there for the long term. And there is a specific dedicated technical support available on that Gridiron Q line. That Gridiron Q line has 5 independent flow cells, as you see on the picture, and each flow cell enables you to run one type of assay, either mRNA or plasmid sequencing. You can run one mRNA per flow cell, so 5 at a time, or up to 96 plasmid at once. So it is a very decentralized, but also a very scalable device. So if we look briefly at the plasmid assay before looking into more detail at the mRNA one, it's a very simple and rapid workflow to perform. The data is basically available within 4 hours. You start with growing your colonies. The library preparation is really quick, in less than an hour, and the sequencing takes about an hour or two, and the data analysis it's about an hour also, and provides you a full plasmid map, fully annotated. It's a very convenient assay, very rapid, and it is nowadays admitted that at least within the research community, about 40% of the plasmid may have some errors in it. So why not doing the full plasmid sequencing for basically the same price, but a very different value? So RNA sequencing, the first publication on RNA sequencing are about 15 years old, and that really enables significant breakthrough across multiple applications, from research, of course, but also biotech, ag, and more recently for drug development. It has really enabled us to better qualify the transcript, identify isoforms that were not needed, detect fusion transcripts that were unknown, or validate them. But if we look really only in the lens of the drug development process, outside of the characterization of drug responses or evaluating the impact of a molecule on gene expression, there is a very clear interest in vaccine quality analysis now that they are mRNA-based. So indeed, as we can perform direct RNA sequencing, meaning we interrogate the native mRNA molecule, no need to do a cDNA. Why would you want to settle for a proxy like a cDNA when you can have access to the original, especially with the base modification? The preparation takes about 2 hours, so it's a very, very quick. You do not need to wait to have dozens of batches of samples to batch to be cost efficient, and you can have your answer very quickly within a matter of a day. So what we have done to start is to build a multi-attribute mRNA QC test. So to piggyback on Michael's slide from previously regarding the traditional method, you do need multiple set of equipment, as Michael was listing. What we're proposing to do, what we have done actually, is combining all of that into a single test that queries identity, integrity, and purity through the main CQAs listed there, from sequence confirmation to poly length measurement. And that enables you to save time and save money also by combining all of the assays into one. And there are already multiple publications on it, and multiple vaccine manufacturers already using this technology and have spoken publicly about it. So how does it look into a little bit more detail? So we start with the product, the IVT, from which we're going to decide to either sequence all of the bases or the bases with the N1 MPUs also. So we start with the poly-A, we add an oligo-DT, we stabilize the structure because mRNA is unstable with a cDNA that is not sequenced. It's just there to stabilize the mRNA. And that takes about 2.5 hours. It is fed onto the sequencer that's sequenced in real time. It identifies the bases for the base calling process. You will have a previously defined a reference sequence. It can be a multivariate reference. It will then provide you all kind of metrics in terms of coverage and variant calling, consensus analysis, and a full report. So the report is highlighted on the right-hand side. So it's a fairly comprehensive quality control report that will have all of the run performance delineated, the different kind of statistics, the integrity. And what we're working on, as was highlighted on one of the slides before, was the development of the 5' cap identification and the double-stranded RNA that will be coming soon, that are under active development. So if we look at that report here, an example of a multivalent mRNA QC, you do get a lot of data points. So you have all of the total reads, the person of identity, very specifics in terms of the poly-A, I mean the mean length, median length, the person present. There is also the, as I was mentioning, the multivalent analysis. So now if we look into more detail in terms of the RNA sequence, it used to be perceived that nanopore quality of the sequence was inferior. It used to be the case years ago. It is not anymore. So what we provide you is a very homogeneous coverage, as shown on the top right here. On the vertical is basically the sequence with each base and the average coverage. The second diagram is the Q-score. And as you can see, the Q-score here is at minimum Q35. So today for mRNA sequencing in that workflow, Oxford Nanopore enables you to reach Q35 to Q45, Q50 even. So it's a very accurate methodology, and we provide you a per base Q-score. We specifically identify the different variants, as shown on the bottom, and their frequency. So you have a very in-depth and precise report. The poly-A tail length as a specific part in the report, in the metric that we measure. We can identify bimodal distribution, the percent of reads that do have a poly-A, and how much are within the expected boundary if you specify between 110 and 120, for example, or something like that. So we basically have developed a very comprehensive end-to-end workflow that is available on the market, and that we are very happy to have our partners like Exotera working very closely with us. Thank you very much. And Sonia, over to you for the poll question.
Well, thank you very much, Olivier, for your portion there. And I will do our poll question right now. And here you go, everyone. Our final poll question. And there you go. Okay, so which stage of RNA vaccine therapeutic development do you see brings opportunity for improved sequencing-based quality control? Okay, so in this one here, you're allowed multiple answers, so choose any of the below. Is it raw material characterization, mRNA integrity verification, process development optimization, or final product release testing? Those are the options that you have, and you can select more than one. So please go ahead and make your selection, and I'll leave it open for a few more seconds so we can get right back into the great presentation with Wei. Okay, that looks like it's half of our audience, so we'll leave it open for the other half to join us, please. Thank you so much, everyone, for your participation. Okay, that looks like it's everyone. I'm going to now close the polls. Okay, hold on. A few more people are joining in. Sorry about that. Go ahead. All right, now I'm going to close the polls, and let's take a look at the results. So here are the results. 42% of our audience said raw material characterization, while 73% also chose mRNA integrity verification, and then 58% for the process development optimization, and then 65% for final product release testing. So thank you very much, everyone, for participating in all our poll questions. Now it's my pleasure to pass the mic and the controls over to Wei. So Wei, when you're ready, you may begin.
Okay, great. Thank you. Thank you for the great introduction, and I'm really happy. Well, Sonia already did a nice introduction to me, so I won't waste your time. My name is Wei. I'm an senior analytical scientist working side by side with Michael Langban in Oxford to try to build this amazing mRNA platform. And thank you for the nice introduction and information already provided by Michael and Olivier. So for now, I'm really pleased to see the fact that people are interested with different aspects from starting materials until the drug products. And luckily, in the following content, I will cover all the different aspects, and I hope you can find the following context to be interesting. So from here, we're inside of Exoterra, we are using this amazing sequencing technology that has been well introduced by Olivier to perform many quality attributes testing for mRNA. The following talks will mainly focus on identity polylens testing, while also data related with integrity, 5' capping efficiency, etc., will also be mentioned. Firstly, I'd like to start sharing the data we obtained using the nanopore sequencing on the quality attributes of the starting materials of mRNA production, which is plasmid DNA. During the plasmid DNA testing, one of the sequencing kit, ligation sequencing kit provided by Nanopore raised our attention because this is a kit that allows us to prepare DNA library without further fragmentation needed. As we can see in the data shown on the table in the left, after tested, we only showed part of the data we obtained, and after we tested dozens of the constructs, first of all, we get one identity result after aligning the consensus sequence we built using the experimental sequencing data compared with the reference sequence. On the tested sample, we get 100% identity. Yet for each sample in the middle column, as you can see in here, we get more than 100,000 of sequence that generated for each individual tested sequence. I would like to mention for this kind of sequencing dips actually is not the limit of our analytical platform or the nanopore sequencing technology. So the sequencing dips can always be increased to further step upon to the client's requirement. So after generating this huge amount of data using nanopore sequencing, we started wondering, apart from identity test, what kind of other attributes, quantity attributes that we can also report using the same data set that we obtained. So first of all, we did some alignment to do the annotation of all the sequences that we generated from each sample. And as you can see on the left column in here, from 83 to 99% of the sequence that ever generated inside of the sample would still be annotated as a plasmid, where for the part there is always a small percentage that couldn't. which can already have some first idea for us to understand the integrity or the possible contamination may existed inside of the tested sample. When we zoom in the results that we obtained with a construct that with really high percentage of sequence could be aligned with the reference sequence, as we can show in here with the sequence length distribution after sequencing. For this sample, we get one single peak that is with exactly the expected target length. Well, for the other sample, this spec sample, which we get a relatively lower percentage of sequence peak could be annunciated as a plasmid. Apart from the peak that represent as the expected length, we also get other different sequences that is not within expectation. In-house, we built different DNA libraries, for example, for E. coli annotation, and so with the further annotation, we understand that the possible contamination that for this P-spec construct will most probably come from the host genomic DNAs. So I would like to mention here, within one sequencing run per sample, we are able to produce identity, result, integrity, and also to predict the contaminant resources inside of the sample. Yet this is not even the limit. Still, using the same data sets that we obtained from this individual sample and using the poly-A tail estimation bioinformatic pipelines on those samples, the poly-A length of the plasmid DNA was also estimated. We observed about less than 5% variabilities, as we can see in the table in here, of the poly length among the theoretical length, the length provided on the COA, and also the length measured by sequencing. We do see two constructs that highlighted in here with a relatively high variability measured between the theoretical length and the sequencing measured by Nanopore. However, within the following test, the mRNA produced using this exact plasmid was also observed with longer poly lengths by Nanopore, as we are shown in the figure on the right. This kind of inconsistent poly length on both DNA and mRNA levels measured by sequencing also shows that this kind of relatively larger variabilities between theoretical and sequencing measured poly length is more come from samples themselves instead of the analytical tools we are using at this moment. After we already explored the plasmid DNA release testing using Nanopore, I would like to explore how these technologies could be used for quality attributes testing on mRNA produced after AVT, which including the AVT mix right after production and up to DS and DP samples. As a long-read sequencing technology with the possibility of sequencing native mRNA, as already mentioned, we selected the direct RNA sequencing kit to allow us to have the most broad and general view of what is really inside of the sample. With this sequencing kit, it allows us to predict the different kind of mutations or structural variation like partial deletion, insertion, and even 5' deletion, as we are mentioning in the figure on the left in here. However, what I'm more exciting is to share this study case that we obtained from a field sample on the right in here. As you can see on the bottom, we have a well, this is really just a partial of the data, but we get a lot of experimental sequencing data generated. After aligning those experimental sequencing data with a reference sequence, a deletion was observed from about 68% of the sequences, while with the rest of the sequence without this deletion observed. Personally, I really think this is a really good example for us to see the power of long-read sequencing, because imagine if you implemented the same sample using short-read sequencing, which First of all, it will be impossible to provide a ratio of the molecules, the percentage of molecules with or without this kind of deletion, or to the worst case scenario, this kind of partial deletion could even be overlooked during the de novo assembly steps if the sequence was generated by short-read sequencing. Another important quality attribute that measured on the mRNA drug substance within the same run as mRNA identity determination is the poly dispersity, as the data we are showing on the left in here. So on the figure in here, I listed the theoretical poly value on the top line with their corresponding measured poly average value on the bracket under them. Of course, we see some standard deviations with a histogram shown in here. However, still between these two values listed, we still only observe the 3 or less than 3 nucleotide difference. And I would also like to mention as with the current process, it allows us to obtain the result from within the same day from launching the experiment till generated the report. With this kind of speed during our in-house IVT process optimization, what we observed is that the different process could greatly have impact on the poly dispersity patterns. And with this current method, it could greatly help us to on the process optimization in mRNA manufacturing in a really fast and cost-effective way. As we already mentioned, that both mRNA identity and poly dispersity are two really important quality attributes for mRNA. With this kind of context, we would like to gain more insight on the method variability, detection limitation, and testing ranges to allow us to provide a better analytical service. With this context, the method qualification was also performed inside of ExoTerra. First of all, as you can show in the table on the left top line in here, we selected one control sample, which is just a generic mRNA with from 5' to 3' with a poly in length with 106A inside. On top of that, in-house, we also designed another SPECT sequence that is 100% identical compared with this control sequence, yet with another 44 nucleotide insertion that is constructed right in front of this 3' UTR. What we have done is to spec this sequence with insertion into this control sequence from 0.5% to up to 1% into the control samples. As the bottom figure showing here, even when we expect the sequence was as low as 0.5% in the testing sample, the current method could still detect them successfully. In the meantime, we also in-house designed and produced another sequence that is also identical with the control sequence from 5' UTR until 3' UTR, just with a shorter poly A in length with 13A instead of 106A inside. What we have done is to spec this mRNA with shorter poly A into the control sequence from 10% up to 100% with 5 different testing levels. On the figure from the top right, we can see that a really good linearity with R square over 0.99 was observed on the whole testing range, and on the bottom right figure displayed a less than 20% absolute variabilities on the whole testing range. So we can say which really achievable performance for both the mRNA identity test and the poly dispersity testing in-house at this moment. As Mikhail already mentioned, in the final drug products, the produced mRNA will be encapsulated into LNP, and during the formulation, mRNA will be exposed to heat and mechanical forces, which could have impact on the mRNA integrity. With this context inside of Exoterra, we would like to understand this kind of how this kind of formulation could impact and to which extent could impact on the integrity of the drug product samples. First of all, in-house we developed a protocol to allow us to release the capsulated mRNA to perform the sequencing test in parallel with the drug substance samples, which is without formulation. As the two data show in here, we can see a slightly decrease of the sequence length was observed on these DP samples compared with DS samples, which could show a slightly decreased integrity during the formulation. With this method, it allows us to provide sequence identity and mRNA integrity data on both DS and DP samples in the same sequencing run. As well, we already talked a lot of data that we obtained using direct RNA sequencing kit, which is mRNA identity, polydispersity, and also integrity for DS and DP samples. But actually, there is another kit, PCR-CDA sequencing kit that is supplying by Oxford Nanopore, and we also give it a try for some of our samples, which with the data generated, we do have some interesting patterns that observed on the left figure showing here. What we observed from the data generated from PCR-CDA is that the cap analogs incorporated during the transcription could impact the starting nucleotide preferred by RNA polymerase. Since we are using the cap AG analogs in-house at this moment, a capped molecule is with A as the first nucleotide, while the uncapped molecule is with G as the first nucleotide. By calculating the appearance percentage of the starting nucleotide, it allows us to also provide the 5-capping efficiency data using nanopore sequencing. Well, we agree that nanopore is still young compared to a lot of traditional golden standard analytical method. It is a cutting-edge technology, but we do want to understand the performance of this new technology compared to the relatively more traditional analytical method. Within this context, as we can show in the figure from left in here, we compare the polydispersity data on the same sample that is measured by Oxford Nanopore, a direct RNA sequencing kit, and also by LC-MS. With the detailed data was shown in the table as below, and we have to be honest, a relatively larger distribution standard deviation was observed from the data obtained by Nanopore, while the data obtained by Nanopore is with an average and a median value that is closer to the theoretical length compared with the LC-MS data. Meanwhile, the 5-capping efficiency on two different constructs were also measured by nanopore sequencing and HPLC method in parallel, as what we are showing here from the right figure. Less than 3% absolute difference was obtained by these two methods. What I also would like to mention is for nanopore sequencing is also a non-construct specific method, which means no construct dependent method development is required upon new construct, which can also be listed as another advantage compared to HPLC method when we talk about 5-capping efficiency. Even though we do see a difference on the value generated by nanopore or other more traditional golden standard analytical method, but we still believe the result is comparable by those different methods, and this do raise the hope that nanopore sequencing could provide more than identity determination as what we are commonly expected from the sequencing method or the more traditional sequencing method as Sanger sequencing or short-read sequencing platform. With nanopore, it allows us to provide multi-quality attribute data to realize the analytical result in a faster and more cost-effective way. After we share about the mRNA data, we believe that even though mRNA has been incredibly successful, especially in the context of COVID-19 vaccine, yet self-amplifying RNA and circular RNA with their enhanced protein expression level, longer duration, increased stability compared to mRNA, starting to raise more and more attention in RNA therapeutic world. With this context, a series of analytical methods has been developed on both self-amplifying RNA and circular RNA inside of ExoTerra, still using the nanopore sequencing technology. For self-amplifying RNA test, what we have developed is identity test, integrity test, polydispersity, and 5' capping efficiency. I didn't put the identity data because it's quite similar compared to mRNA. We align the experimental data with the reference sequence, so there is not a lot of mystery in here. Yet, as we can, we started from the integrity data that we generated, as shown on the left of this slide in here. Compared to the integrity data that we generated by fragment analyzer, which is about 80%, the data we generated for integrity by nanopore is only about 71%. We do realize that with a relatively larger size of the self-amplifying RNA, there is a higher chance to bring the sequence to further fragmentation during the multiple steps of sample preparation. So at this moment, we are also doing a lot of optimization in-house, try to solve this problem and try in one day to provide the integrity and identity data for self-amplifying RNA in one sequencing run, as we are already doing today for the mRNA. When we look at the middle figure in here, the measured average poly length, which is 106A, is only 3 nucleotide difference compared with the theoretical 106 in here, which also indicate a really good confidence using this method for polydispersity. As shown in the right of this slide, we also observed a similar cap analog incorporated during transcription could impact the starting nucleotide preferred by RNA polymerase with similar pattern as we already described as mRNA. With this context, we can also provide, even though the cap analog is slightly different between the self-amplifying and the mRNA, but we can provide 5' capping efficiency data as well. When it comes to circular RNA, first of all, we know that with the name already tell us the format of circular RNA is circular. So first of all, in-house, what we have done is to develop an in-house treatment protocol to transfer the circular format into a linear format, and with a polyetail added to allow this sample is compatible with the direct RNA sequencing kit supplied by Nanopore. I would like to mention that for this in-house protocol, there is no amplification steps, which means there's no bias that introduced during the sample preparation. We are still able to sequence what the sample really is. So with the prepared protocol and the direct RNA sequencing kit, we are at this moment, we are already able to provide identity determination for circular RNA sample, as we can see in the right figure in here. We can see a decreased coverage indicated in blue from 3' to 5', which is a similar pattern observed from mRNA sequencing since the sequence is starting from polyetail. Meanwhile, as we can see from the left figure that currently we need to be honest, this method could still cause some fragmentation during the sample preparation step. This part is still under optimization, and hopefully we can use the data generated from a single sequencing run to provide identity, integrity, and even circularization efficiency on circular RNA in the really near future. After sharing all this data related with different types of RNA, I would like to mention that as a CDRMO, ExoTerra is also delivering preclinical and clinical process for not only RNA, but also various therapeutics. To further explore and widen our analytical capacity, related analytical method using nanopore sequencing was also conducted on adeno and adeno-associated virus. As part of, you may know that adeno-associated, also known as a virus, also known as AAV, is a single-stranded DNA virus, which is not directly compatible with the double-stranded DNA sequencing library preparation kit supplied by Nanopore at this moment. So the first thing we have done is to develop some in-house sample preparation to allow and still without any amplification step to introduce bias to allow this kind of single-strand DNA format is compatible with the ligation sequencing kit. And as we can already see in here, a really good integrity has been obtained and shown as on the top left figure in here. Also, as a long-read sequencing technology, nanopore sequencing offers the capability to sequence the full genome of AAV from ITR to ITR without any fragmentation of the DNA molecules. With this kind of capability and the DNase-1 treatment before DNA viral extraction, the current method also allows us to provide information on the ratio of fully and partially packed AAV by analyzing the annotation of the sequence, the length of the generated experimental data, and the number of the sequences generated after sequencing. As what you can show in here on the bottom left figure in here, within the same sample using nanopore sequencing, we get about 55 to 60% of fully packed AAV ratio, while this same sample was also evaluated by another classic analytical method, analytical ultracentrifuge on this sample, and about only 3% absolute difference was observed on the percentage of fully packed AAV. Last but not least, our in-house constructed DNA libraries could also provide DNA contaminant sources annunciation, as shown in the figure at right in here. Apart from the 97 to 99% of the sequence could be directly annunciated as an AAV, we can also predict and provide the ratio of other contaminants as the plasmid backbone, helper RepCap plasmid, or even to different kind of host cell DNA that we can construct it in-house upon to the need. So that's about all the data I would like to present, and I would also like to take a step back with you to look at the current USP guidance that already been mentioned by Mikhail earlier. And as for today, a bunch of important quality attributes mentioned by USP guidance are realized with different kind of analytical method, which also including really heavy machines like HPLC, MIS, fragment analyzer. However, when we look at the data we already see today, a lot of them, the test from starting material until the drug products can be realized on this one single Gradian equipment. And with the network of Nanopore to have this Gradian Q-line available, now we have the test that is ready for both R&D and QC environment, which is really a good news and a great increase of the capacity for us as a CRDMO. So some summary about the presentation for today. As in Exoterra, we are able to provide analytical method development from starting materials to drug product samples, which is also available in both R&D environment and QC testing. We have really complete testing, analytical testing panels for both RNA related and viral vector related projects that tailored to the requirement of the client. With the expertise we have, we can also develop some in-house sample preparation protocols to suit for the need and for the different sample types as new as to self-amplifying, RNA and circular RNA, or as classical as to AAV or ADV. We also provide a bunch of different customer requirement analysis, like to build different DNA or RNA libraries for annotation usage, or to vary our sequencing tips just to the need. Really, the last, I would like to extend my thankfulness to the amazing team I'm working with, not only in Exoterra, but inside of Oxford Nanopore. You are the person who make all the data that present today and to make this presentation come true. Thanks a lot. And now I will pass the microphone to Sonia again. Thank you.
Well, thank you very much, Wei, Olivier, and Michael for that great presentation. I think we have time just to squeeze in one question. So remember, audience members, you can still send in your questions now using that questions window. We won't have time to answer it today, but we will get back to you after this webinar is done. And for those of you watching on LinkedIn Live events, please feel free to post your questions on the comments section. And let's just squeeze in one question here, okay? This audience member is asking, Nanopore could generate large amounts of data as a CDMO. How can Exoterra is sharing the size of data with the client? Who would like to answer that one?
If Michael don't mind, so I will take the lead. Actually, at this moment, we are also using clinical data safe security level of the server or of the cloud-based server to store all the database, and in the we are also under discussion to open the portal function to allow us sharing the data based on this server with our potential client in the near future.
Okay, and I'm going to squeeze one in for Olivier. Does the mRNA QC analysis, including poly, already available on Epi2Me software, or will it be released soon?
It is available as part of the end-to-end solution in the mRNA quality control kit, and it's only available on the Gridiron Q, which runs completely offline.
Okay, thank you. We squeezed in two questions. Well, thank you very much for those answers. We have reached the end of the Q&A portion of the webinar. And as I mentioned before, the team at Exoterra will follow up with you for the questions that we did not answer. And if you have any further questions, please direct them to the email address on your screen. So it's info@exoterra.world. And then there's a phone number there, exoterra.world. And then if you have any further questions for our panelists, our speakers, please go ahead and utilize the email addresses there. And then you notice there's a QR code. Please go ahead and check that out. I will take you right to their websites, okay? So thank you very much, everyone, for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated, as it will help us to improve on our further webinars. Give me a few seconds, and I'm going to share a link to view the recording of today's event in the chat box, which you can also share with your colleagues once they register for the recording here as well. And for those of you watching on LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and gain access to features such as viewing the recording and more. And as I mentioned before, I told you about the QR code. So the only thing left to do right now is say thank you very much to Olivier, Wei, and Michael for their time here today. Thank you so much. Thank you. We hope you found this webinar informative. I know I did. It has been a pleasure to be your webinar moderator. On behalf of the team here at XTalks, we thank you for joining us. Until next time, I'm Sonia Hunt. Bye for now, everyone. Bye.
Bye.